Hello, everyone. Welcome to the CyVerse webinar series. I'm Tina Lee, CyVerse's User Engagement Officer. Today's webinar is a much requested follow-up from a webinar that he gave in the fall. Dr. Jacob Landis is our presenter, and he had done one on SNP calling to identify variants last fall. And today's webinar, he'll demonstrate how to use CyVerse apps to do downstream PopGen or evolutionary analyses on those variants that were identified. For those of you who are new to CyVerse, we're a cyber infrastructure project funded by the U.S. National Science Foundation. This free webinar series helps fulfill a key part of our mission to train scientists in how to use CyVerse's computational resources. First off, we'll do some quick housekeeping. Today's presentation is approximately 30 minutes. Please mute your audio, but do enable the chat window so you can type questions there for Jacob to answer after his presentation. We'll also post any important links there for you to find. And this webinar will be recorded and materials will be posted on our website later on this week for you to review at any time. In addition to these webinars, CyVerse offers trainings and most of our learning materials are already online. So go to our website, CyVerse.org, check out the learning pages for more information on upcoming events, including workshops and trainings that will help you learn and teach using CyVerse. And now I am pleased to introduce Jacob. Jacob Landis is a lecturer for plant systematics at Cornell University in New York and a research associate in the SPECT lab. He also works closely with Susie Strickler and her group at the Boyce Thompson Institute's Computational Biology Center. His research interests are primarily at the intersection of evolutionary genomics, phylogenomics, and population genomics. Jacob has been instrumental in optimizing the Oxford nanopore technology for both DNA and RNA for a variety of monocot sequences in Dr. Speck's lab. So welcome, Jacob. Nice to see you again.
Thank you, Tina, for the introduction, and thank you all for joining us this afternoon. So I'll go ahead and jump right in. As was mentioned, this is a follow-up to last September, a webinar that I gave where we actually went through to call variants using a variety of programs called Stacks and GTK. That webinar has been recorded. It can be found on the CyVerse website. I put a link down here. Today, basically, we're going to pick up from where we left off. And we're going to cover some of the downstream analyses after you have your variants called, after you've done some initial filtering, and you are happy with the data set that you have. Data and scripts have been posted on GitHub. There's a link here. It will also be posted in the chat. There are two different tutorials there on GitHub. One is with the CyVerse discovery environment, which is what the main focus will be today. And the other one is a more command line driven example for most of these same programs. And I'll give a reason why in a few slides about why we have these two different tutorials. So I mentioned the CyVerse discovery, and on the slides, you will see this designated here with the CyVerse logo. But this discovery environment, once you log into CyVerse, and there's resources on the tutorial as well as other places on how to actually access this. But for almost every analysis that we're going to cover today, and that's in the tutorial, it has its own app. Though many of these analyses will be using RStudio, and so we'll be using the same RStudio instance. It already has everything loaded that we need for many of these analyses. However, with these apps, they're limited sometimes in some of the options we can have. So most of these programs have many, many more options included in them than the apps do. And so if you want to explore some of these analyses more in depth, I would suggest using the command line versions on local machines, local servers. But these apps will be a good way to get started and will do most of what you will need. So what can you expect after this? As I sat down to try to create this webinar and go through the tutorial, I realized that it's very hard to do this to encompass all skill levels. So when this is all said and done, I do not expect anyone to be a pro at any of this. But my main hope is to decrease the learning curve for your own projects. So here's an example on the right. Maybe when you're first starting to figure these programs out, it takes a while to figure out what the input outputs may be. I'm hoping that after this webinar, we can really increase your progress so that you are doing analyses both with this test data set and your own data set to start addressing questions that you're interested in. However, I have been able to break this down a little bit more about what you might expect with different levels that you are interested in. So if you have never done any of these analyses before, we have with the tutorials, we provide workflows with CyVerse, particularly or specifically the discovery environment and other standalone programs that will allow you to go through and do analyses that you want to start generating plots for publication or talks, and have everything contained right there. If you do have some experience though, but you want it a bit more, in the command line tutorial that's on GitHub, provide additional examples, and then you have a bit more flexibility in some of the flags and other options that you call. So you have a bit more power there with some analyses. And then if you are an advanced user, I'm hoping to provide some alternative methods and other avenues to explore, kind of more with your own data set. But hopefully, no matter where you sit on the spectrum, there will be something useful for you. So what is it we'll be covering today? We'll be covering quite a bit. Some of these topics include tree building, looking at how your samples are related using PCA or structure, looking at isolation by distance, and also some genome-wide association to look for association between SNPs and phenotypic traits of interest. So there are many options available for each of these different topics. We will cover just some of those, and I'll highlight some other examples that you may want to look into more. And throughout the webinar and the tutorial, try to highlight the fact that some of these analyses work better for particular data sets. There are different things to keep in mind, computational requirements. If your SNPs were called with a reference or a de novo approach, that will impact some things. The number of contigs that you have, if you have a reference genome, is it high quality? Is it kind of more of a draft? All these things can work, but they will change a little bit about what the steps are that you need. And I'll try to highlight these when possible, both again in the slides and the tutorial analyses. Now, before we get to what you all came here to hear more about, I do want to say thanks to those who have helped with this. So throughout this webinar and the tutorial, you'll see apps in the discovery environment for many of these analyses. And you'll notice that there are two particular names that show up a lot. Ritu and Amanda, both from CyVerse, have spent a lot of time helping me get everything set up and running and optimized operational for doing these analyses. So I want to personally say thank you for the amazing work you've done to help out. And hopefully when you all are using these programs, you can also thank them as well. One last thing, the data that we're using today in this webinar and the tutorial is a modified variant call format file that was as part of a recently accepted publication on the Washingtonia palms. This project was led by Lorena Villanueva. In that study, we had 181 accessions where we looked at population structure, relationships. However, in a lot of these analyses that we're running for the tutorial, we have pruned that down from the original data set down to 48, basically for ease to make things run faster. With this data set, we know where the populations were sampled. We know geographic locations from those. And we also have some phenotypic data as well as some simulated data that we can use for the genome-wide association later on. However, this was not included in the original study. This paper has been accepted, the proof's been submitted, but it's not online yet, but it should be in the next couple of weeks. And that's from the Botanical Journal of Linnean Society. If you want to see how this all fits in, check it out. Also, if you want to go grab the raw data that's associated with this paper, the links are in there as well. So that's the setup of what we're gonna be doing. We have the data, we have our platform. Now let's jump into doing some actual work here. So the first thing I want to cover is file conversion. And if you have done any of these analyses yourselves, you know that file conversion is a major pain. Most of these programs do not take the same input files. So you have to convert them from one format to another. So here are 4 examples I'm showing. This is all basically the same data, just in a different format. We have our standard VCF or variant call format, where each column is a individual and each row is a SNP or a locus. We have our structure format, which we'll need today as well. In this case now, each individual is represented by 2 lines, and each column is a locus. We have our FASTA file, which we'll use for some of the tree inference. Here, we just have a string of nucleotide sequences. And then for gene pop, which we'll use for isolation by distance, you can see that the format structure is quite a bit different. But again, all of this is the exact same data. It's just been formatted differently for different inputs. So there are a few tools that exist to help with this problem. We'll cover a few of those. There's many more out there. The first one, though, is we can use stacks to help create some of these files. So stacks we used in the earlier part, or for the first webinar, to call SNPs, either a de novo or reference guided approach. However, it has built-in functionality where you can read in a VCF file that you have already filtered, you are confident in your SNPs, and it will create different downstream files for you. All you need for that is a VCF, your variant call format, and a population map. In this particular case, my population map is just for each individual being its own population. Some of these outputs do it on a population basis, some are individual basis. So it is probably worth exploring that for your own data if you want to. But everything we're doing here is based on the individual. So within the discovery environment, we can use the Stacks 2.53 populations app. We'll specify our VCF, our population map, and we can create our downstream files. If you're doing this on the command line, this is essentially the code there. You're calling the populations module in Stacks. You're giving it a VCF file, telling it where do I put these files, giving it that population map, and telling it what you want to export. In this case, VCF, gene pop, and structure. And if you look at this tutorial later on, you can see exactly how you do this in the discovery environment. And so this will create most of the files that we want. Some of the additional ones we might want are Nexus and FASTA files for some of the tree inference. There's a different program that works really well with that. Actually, it's a Python script called VCF to fileup. All you do is you give it your VCF file. In this case, you don't need to worry about your population map at all, and it will take that VCF file and create, in this instance, a Nexus and a FASTA file. Again, this has its own app in the discovery environment. If you're doing it on your own computer, here's the code that you want to use. So now we have our files. Now we can start thinking about what analyses do I want to do? What do I want to show? And if you're interested in showing relatedness, which we all are to some different degrees, there are many different ways to do this. However, again, the input is different if you're doing a PCA, or if you're doing a structure plot, or if you want to infer phylogeny from different ways. Both the input files and what you would need to accompany those may be different. For PCA and structure, you probably want to have some prior knowledge on how the sample is grouped together, but it's not necessary, but it can change how you can visualize that and how easy it is to read the output. One thing I haven't mentioned that we talked about though in the first webinar is pruning, in this case, especially for linkage disequilibrium. And that's important in this case, especially for the phylogeny aspect. You want to keep SNPs that, or you want to remove SNPs that tell the exact same story and are always inherited together. If you leave all those SNPs in there, especially for phylogenetic inference, it can inflate your bootstrap values to above what you should normally see. Additional piece to keep in mind with linkage is that it also, if you prune for LD, you can also speed up many of the analyses that you're running because you have fewer loci to keep in the data set, and those loci tell the same story, so why keep them? So if computational resources are important, definitely prune for linkage will help in a lot of these aspects. So for comparisons, first up, talking about building a maximum likelihood tree. We can do this in a program called SNP Philo. Again, it has its own app. A few things with this one is that one very important aspect of this program is that the chromosomes or the contigs cannot have any letters or words in those. It can only be represented by numbers. So if you have a VCF file that has contig 1, contig 2, that's going to cause some problems and cause it to crash. So we need to remove those. Additional thing with SNP Philo, the default for this is to only have 22 chromosomes. Now, if you have a high quality reference, that may not be an issue. If you have a draft reference or a de novo approach, that can cause some problems. However, there's an easy way around that, but we can tell the program how many chromosomes to include, or what's the last autosome in the data set. And so one way you can do this is just give it the arbitrary, say there's 600,000 chromosomes here. This will not cause any problems in terms of analyses, really. It does impact a little bit the linkage if you're pruning that in this program or not, but it will run. In this case, for our data set, we've already have pruned the data, have high confidence, pruned linkage. And so here's an example of the code that if you run this on your command line, I have set LD to 1 by this L flag because we've already done LD pruning, though you could do it yourself. When we let this run, it runs fairly fast and we get a phylogeny similar to what we've seen before. In this data set, we have 3 replicates per population. And you can see that if you can look closely, a lot of our populations are matching, they're closely related to each other, so we're pretty good. However, building a phylogeny this way with SNPs and maximum likelihood may not be the best approach. Basically, we're using a concatenated data set, so we may have different signals in our data set. And if we're doing populations, those populations may have gene flow, and so that breaks one of the assumptions of phylogeny of a no hybridization. So maybe this maximum likelihood approach isn't the best. So one option is using a Bayesian Coalescence approach in SNAP. This can explore the conflict and signal between SNPs. However, I want to make it clear it is very computationally intensive and can be very slow. And so with large data sets, this is definitely not a good way to go. Even if you group things by population, it can still take a long time to run. One additional thing is with this method, it ignores loci with missing data completely. So when I run this, I usually prune down further. This is with VCF tools where I allow no missing data and also thin it out so there's more space between each loci to reduce our data set from, say, 20,000 SNPs to, say, 3,000 or 4,000. Once you have your VCF, your pruned down VCF, you will create a FASTA file, which is the input, which you'll use to generate an XML file in Beast2. Now, this part will be done on your local machine. Beauty is packaged with Beast2, so you can run this on your own. It does not take any resources really at all to do that. Set up the XML file, and then you can run it on Beast2, either in the discovery environment or on your own computer. Again, here's some of the pipelines for that, or the apps that you'll be using in the tutorial itself. So once you have that, so here's some screenshots from Beauty. So I'll input my FASTA file. I'll have my taxon identifiers. I'll have then group those based on species or populations. In this case, again, we have three taxa per population. We'll set that. We'll tell it to calculate mutation rate based on the data set I give it. And then for this particular run, we'll run it for, say, 100,000 generations, which will probably not be long enough, but we can at least see how it looks within the discovery environment. You can then run that with Beast. However, we did notice a problem yesterday where some of the files weren't being written appropriately. So maybe, for at least the time being, kind of ignore Snap. Again, it is very slow compared to other things. However, if you're going to run this on your own servers, your own program, or your own computer, you can call the Beast, the Java file of Beast tell it how many threads you want, and you can just run this XML file, and you'll get a phylogeny very similar to here. So this is a species tree inference. You can see there's multiple branches in some cases, so some of the SNPs are showing a different pattern than others. So it can be very informative and very helpful, but again, it is fairly slow to get running. And it'll show some differences, but this may not be the best approach, especially if you have lots of hybridization or if you have lots of gene flow between your populations. So another maybe better approach is using the NeighborNet strategy and splits tree. So this is a distance matrix approach versus Bayesian or maximum likelihood. However, the really helpful thing about this program or this method is that it does not rely on a purely bifurcating or hierarchical layout. And so you can definitely have things that have conflicting signals. And it's a collection of clusters represented by splits graphs. And then some of these splits may show conflicting signals. However, in what they call ideal data sets, you'll get a tree-like or a tree pattern. This is also very effective for computing hybridization and recombination networks. So here's an example from a few years ago where they use this approach and your traditional phylogeny. So you can see the traditional layout. However, if you look at the NeighborNet, you notice it's not that clean, it's not that easy. And with the structure analyses too, you can see that some of these different groups have more gene flow between those. So maybe this NeighborNet is a great way to go. This is a standalone program. It does run pretty fast, has a graphical user interface. There's two different versions, Splits Tree 4, Splits Tree 5. You can go to here and it's on the tutorial as well and figure out which one works best for your computer. Essentially, you can just open up the GUI, file open, give it your Nexus file, and it will create a pattern like this. And so this is in comparison to what we saw earlier with SNAP and the maximum likelihood tree. In this data set, we know that this ALA population is actually probably of a hybrid origin. And this NeighborNet really does highlight the fact that these are probably multiple origins of this hybrid population. And so you can kind of get some more inference here that you'd miss out on other approaches. So these last three have been all tree-based, phylogenetic inference-based, but there are other methods that do not rely on a phylogeny. One of those is the principal component analysis, the PCA. So this is a way to take your high dimensionality data, so maybe tens to hundreds of thousands of SNPs, and reduce this down to something that we can see easily on the computer screen and something that is informative. And usually it's reduced down to the first handful of principal components, also known as PCs. So PC1 is often the direction that maximizes the variance of your projected data. You do not need to designate ahead of time what your populations are or what your clusters are for the analysis to work. However, if you do give it population designations, you can color code these things to make it easier to see. And then one last point I want to make on with the PCA before I show some of the results is that the top PCs of a genetic PCA, so in this case with the SNP data, often explain a much lower percentage of the variance than the top PCs from a phenotypic analyses. So if you're looking at phenotype data, you may think the first couple PCs should be 70 to 80% or explain that much of the overall variance. And that's probably very true. With genetic data, if you get 20 to 30% of the variance explained by the first couple PCs, that's actually really good. And part of the reason for this is just the sheer number of characters. Phenotypic data, maybe you have 10, 20 characters. With the genetic data, you're talking, again, tens to hundreds of thousands of loci. And so, the amount of variation that each trait may explain is going to be small. So within the discovery environment, we can run this principal component analysis with RStudio. Again, all of the libraries that we need for this and the subsequent RStudio analyses are already loaded. You will launch a web browser for RStudio, and then you can run through all the commands as you would on your own machine. But again, you're using some resources from CyVerse The scripts I have in the tutorial are all meant to run the analyses and write the PDFs of each plot so that you can view these when you're in your RStudio instance. If you go to File, Open File, you can select a PDF and see something just like this. And then you can download it, save it to your local machine. Makes it real easy to get the output for you to do what you need to. From this particular script, it's going to run, it's going to create the principal components for the first 4, 1, 2, 3, and 4 PCs, and plot different groupings of those, different comparisons, as well as give the percent of the variance explained for each. And in this data set, we have 5 populations we knew ahead of time, all color coded so we can see how they relate to each other. I want to move from a PCA to a structure, and it's very similar to see how your different groupings or your different samples are related to each other. And there are many ways. So there are many ways to do this, but one way is with the LEA package in R. It investigates the ancestral populations, which is that K value, and mixtures between samples. However, when you're looking at this, you have to be very careful. There are ways to overinterpret the best K value. Here's a publication from 2018. There's a reference list at the end of these slides to look some of these up to say kind of how not to do this. Again, also picking what the best K is not always so straightforward. There's a method called delta K, which can do it. However, there's a strong tendency to pick a K equals 2, which often is not correct. And then this LEA package uses a cross entropy criteria to select the best K. When I show the graph on the next slide, it's the low point of the graph. As opposed to other options, structure, admixture, this package doesn't rely on some assumptions such as no genetic drift, Hardy-Weinberg, or linkage equilibrium. So if you have, this is very suitable for inbred lineages such as plants, it can be very helpful. And then Stacks creates the input we need for structure. However, the file that created by Stacks, we need to delete the first 2 rows of the header in this file. This is mentioned more in the tutorial and it's already been done. Otherwise, the program has trouble reading it. But when you run this R package or this R script, again, in the RStudio, it's going to test for the best K from 2 to 25. So again, for this cross entropy graph, we want that low point. So that's a K equals 4. And then the script will also write or create the structure plots that we're often used to seeing. This is an example from K equals 4 with different colors. If you need to you can move samples around, shift things to make it look easier. You can do that in just a text editor by changing the order of the structure plot. You can copy paste things if you want to make it a bit cleaner. As again, that's 2 lines per sample. But in this example, one's just orientated by population, it looks pretty clean. One additional thing, so now we've seen how populations are similar, how they're different, but why is that? And so one way to look at that is isolation by distance. And so this is a consequence of limited dispersal. Depending on which organism you work on, it may be more relevant or more prevalent than others. And so basically what we see is that pairs of populations that are close to each other are more genetically similar than populations that are far away. However, some environmental barriers can inhibit dispersal, which have a very interesting biological story to tell. And so it is definitely worth testing for this and seeing what happens. However, it's been shown that the strong presence of isolation by distance can bias other downstream analyses, such as outlier loci scans, where up to 30% of the outlier loci are caused just by the spatial patterns. So testing this is a very good thing to do. Again, we can use an input file from Stacks, the gene pop input. However, we do need to rename this as a .gen file instead of the .genpop. This has been done in the example data as well. Then one thing you need to do, this is again all in RStudio, but you need to give it a geographic location file where we have each of our samples. We have our latitude, we have our longitude, all in decimal format. You give that to this program and you'll be able to run through the script and produce a few plots like this. So within the script, it's also doing a 10,000 permutation test for significance. So it has our two data matrices. We have our geographic distance, our genetic difference, our distance, and it's going to shuffle one of those, keep the other one the same, and see what story do you have. And so it produces two plots. One is the frequency or the significance test. So for our particular isolation by distance test, we see an observed value of 0.42. However, the permutations are all much lower down to here. So we have a significant, a very low p-value. And you can also plot this, the geographic versus genetic distance, and you can see in this case that we do have a slope where populations that are individuals in this case actually, that are more close to each other or close to each other or more genetically similar to each other. There are a few alternatives to the traditional isolation by distance. One of these is called Construct. This was released in 2018. This helps model continuous patterns of differentiation, which can cause problems with these analyses. Here's just some examples from the paper in Genetics 2018, showing your kind of traditional admixture model to this construct, either including spatial or non-spatial data, and it can make a difference. But you can also go beyond just geographic distance. We can also look for isolation by environment or isolation by resistance. So IBE is looking for associations or patterns between genetic distance and environmental distances. So you can download the world climb data to use that. Or this IBR, this isolation by resistance, is the genetic distance and resistance distance, which better accounts for habitat heterogeneity. So I'm not going to the tutorial to show exactly how these are done, but they do have a very similar framework to your isolation by distance. And then genome-wide association. This is one of the last kind of major topics that we're going to talk about today. People have a lot of interest in. So for this one, you're going to scan through your data set and look for markers so you see for association between SNPs and phenotypes that you have measured. Some considerations to consider for this, depending on what your phenotypes are, is likely you want to normalize those or transform those in some way. So you have a normal distribution. Having quantitative continuous data makes the GWAS run easier. There are some ways that you can maybe use it for qualitative data, but those don't have as much power. Make sure your sample size is large enough and you have a fairly dense sampling of SNPs. Not really talking about millions of SNPs, but you want to make sure that your SNP sampling is fairly well covers the genome. And then most of these methods are designed for reference genome data. So you have a low number of contigs or chromosomes. Some of these you can use the de novo aspects for that, but then there's also issues with linkage disequilibrium and lack of coverage. And pruning for LD is important. It makes the run more efficient, and it changes the number of SNPs or number of tests that you're doing, which changes your level of significance for where your value is. So with genome-wide associated, there's a few models that people often talk about. One is your general linear model. And so this is basically an association using least squares fixed effects linear model. You can reduce false positives in this case by adding in population structure, such as results from a PCA. There's also the mixed linear model. So this incorporates both population structure and kinship between samples to reduce false positives. However, with large data sets, this can be very computationally challenging. And this kinship matrix that we mentioned is basically just a relationship among individuals. So here's an example, a test data set where the different colors show how similar these different individuals are to each other. And it's broken down by a probabilistic estimate that a random gene from one subject is identical by descent to the gene in the same locus from a different individual. And so it's important to incorporate this to make sure you do not have false positives. Two ways that we'll just briefly talk about how to do this. One is with Tassel. So this is going to be, again, on your own local machine. It's a graphical interface, point and click options for almost everything that you do want to do. It does have a very extensive tutorial online. It works fast and it works for almost every platform. And one thing I like about it, especially when you're first getting started, is that it is not picky about if you have samples missing in your genetic data or phenotypic data, if they're in different orders. It doesn't really care at all, and you can just give it a VCF input instead of having to change it to a different format. One drawback is, though, there's not much control over plotting for your plots, your Manhattan plots, but it will plot something for you. And so here's just a couple of screen grabs from Tassel. So you'll read in your data, and this is all in that tutorial walkthrough. Read in your phenotype data, read in your SNP data. You can go through, you do the general linear model, the mixed linear model, and create a Manhattan plot where each chromosome, in this case, is color-coded. However, one drawback here is it doesn't tell us where the level of significance is. So which of the dots or these SNPs are significant? It doesn't tell us that. However, the next method that we'll look at can. So the next method is MX. This is widely used for larger data sets. However, some of the newer publications are coming out are using a program called Gemma. It works in a similar fashion. One thing about both MX and Gemma, you need to reformat your VCF file to a Plink format. And Plink can be picky, or it actually is very picky about the number of chromosomes that you have. But you can, if you have more than 22, which is what Plink as a default is, you can give it a flag called allow extra chromosomes, and you have no problem. With MX, you have the two steps for the genome-wide association. The first is to generate a kinship matrix, and then the second is to do the actual tests. However, this program will not plot for you, but you can import the data from RStudio or import the data from MX to RStudio to plot, and there's a script to do that in the tutorial as well. In the discovery environment, so again, we have 3 steps now. One is we're going to reformat from VCF to Plink. We're going to have it run the whole MX package in one app. So it's going to make the kinship matrix and do the association. However, it will not write the kinship matrix, unfortunately. Once you have the results, you're going to save those, import those into RStudio, and then using the R script, you will plot a graph like this, where I have each chromosome colored just an alternating grays, but you can change that, and where the level of significance is, and where those significant SNPs are. So for this data set, we have 2 significant SNPs. This whole pipeline, though, is fairly is customizable to what you want to do with your data set. It's just kind of an example of what we can do. So I know I covered a lot of material. It seems like this happens a lot with these webinars, but you can go back to it. You can look at the tutorial to really get a very good feel for how to go through some of these analyses, both in the discovery environment and also in the command line version. So, we've covered many different aspects to get you started. Tree building, population structure, or structuring, or genome-wide association. Almost all of these analyses can be done with a variety of data. This data set that we're using for this tutorial is all from RADseq data. However, HiveSeq, RNA-seq, genome resequencing, all of this will go into or can be useful for all these analyses. The one place where you might have issues again is with the genome-wide association and how many contacts you have, but you can at least get started and start playing around with that. Again, some analyses, though, have special requirements, specific contig, chromosome formatting, or numbers, which can cause problems. And then, again, all these analyses, except for the ones that are on your local machines, we have point-and-click options in the CyVerse discovery environment to get you started, as well as command line options that are on GitHub if you want to take it one step further and really start exploring what some of the options are and what fits your data set the best. These slides are all available on GitHub as well. And here is the references for a lot of the studies that I cited throughout the talk. Definitely, I encourage you to go back, learn more. I just had a kind of a broad overview of some of these different topics, but you can spend a lot more time talking and looking at these. But I just want to get everybody kind of started. And then lastly, I just want to thank CyVerse for having me come back, do this again. I hope will be beneficial, both the SNP calling and now these downstream analyses for your work. And hopefully it will really help increase your confidence and your ability to analyze your own data and to start addressing the questions that you find the most interesting. I know that was a lot. If there are any questions, I can try to address some of those now. On GitHub with the slides, email address, Twitter, feel free to reach out to me if you want to as well. And thank you all for joining today.
Thank you, Jacob. Yes, of course there are questions. You covered so much, and I love the fact that you really give hints about things, right? You're pulling on your expertise and the kind of thing you often don't read about in documentation. So let's get to the questions.
Yep.
From your previous talk, someone has polyploid data and has used VCF tools rather than BCF tools for some of the basic filtering. Do I need to redo the filtering of my original VCF file before attempting some of the analyses you did today?
So, this is kind of a more specific question. However, BCF tools is going to work much better for polyploid data, and you'll be a high confidence in that. Some of these analyses, the assumption is, especially for say the PCA, that you have biallelic data. So you only have 2 alleles. Now in some polyploid data, that might be actually true. And others, depending on if it's an allo and how recent, it may not be. And so I think you could probably get started with some of these analyses based on what you have. But it's also very important to look back and look into some of the assumptions for these different programs and what data is going to be used, and also how confident you are with your genotype calls. And this is true about diploid, polyploid, whatnot. You want to have high confidence, especially if you have a very heterozygous study system. You want to make sure that you're confident in what you're calling is a correct genotype.
Got it. Okay. Next question, is there a minimum limit for the number of transcriptomic samples to get a good SNP calling?
You could technically do it on just one sample. But for overlap between different individuals, there's not really a good threshold. The one key thing, I think, more than just the number of samples, is the depth of the read depth for coverage, and make sure you have enough. But you could call SNPs on 5, on 5 samples. The earlier tutorial we had, I think it was 15 samples. This one we were doing things with 40. So really about anything, the key thing is just to be confident in what you're calling genotype wise.
Okay, next question is about, I guess, the app called Structure. This person has a file from iPyrad and wants to know if they need to also delete or edit the file before running Structure.
So I haven't looked at iPyrad in terms of what the output is. However, I will say if there's any kind of header information that can be deleted. In the R script that I have in this tutorial, you can tell it how many header rows you have, but I find that doesn't often work correctly. The key thing is that, again, with Structure, the basic format is 2 lines of data for each sample. Anything else you don't really need. So if there's any headers at all, you can delete that. But again, I haven't looked at iPyrad, so I'm not entirely certain if it will work. But deleting headers is never a bad thing for some of these programs.
Okay. Next question, do we need to remove related individuals to run the LEA package like is needed to do when running Structure?
To remove related?
Remove related individuals in order to run the LEA package.
I'm not entirely certain about what's being removed. My initial assumption is no, but I would need a bit more detail to be able to accurately answer that question. Sorry for that.
Okay, maybe Karen can type a follow-up later on. Next, if we are working with haploid organisms, how do we resolve the chromosome formatting? Is it only using one input or input one?
Yeah, it's actually a great question. I mean, it's a complicated question, and it's a toss up. It's like the other side with the polyploid question. Some of these programs you can specify what the ploidy is. So for structure, the LEA package, I believe you can specify is it haploid, diploid, or whatnot. With the PCA, I'm honestly not sure. My guess is that you can specify that as well. A lot of these programs, maybe not the ones I've focused on as much, but others will be very useful for haploid data. And so I think just to make sure that the inputs are specified. This is one case where running things on discovery on the discovery environment may not be your best option, because you may need to add additional flags that are not standard. For the R scripts and the R analyses, that you can modify those real easily. So that should be there, but others, I would look into the program to see if you can specify a ploidy. In most cases, you can.
Okay. Can the tools you talked about today be used for bulk segregant analyses, particularly based on RNA-seq data?
If you have SNP data, probably so. I know there's some new packages and new approaches with bulk segregant analysis and pool-seq data that might be more appropriate. So I think you could probably give it a try, but I'm not entirely certain how well it might work. Some of these, like the trait inference especially, might be very biased, but structure, PCA, those analyses, I think might work, but again, I'm not 100% certain.
Okay, and last question. Is there a way to identify the 2 SNP, for example, on the MX figure?
Yes, and so in the R script that's there, I plot the Manhattan plot, but it also creates a text document that writes out each SNP and what its p-value is. So you can download that, open it, and sort it, and then it'll tell you based on the p-value what chromosome and what base pair that is. If you have a bunch of them, that might not be the best approach. However, in R Studio, you could plot it to have an interactive plot where you could hover over it and it'll tell you what that is. I did not do this in this particular case, but it can be done and modified fairly easy with the Plotly package. But the data is there in the output where you could identify exactly what that variant is.
Jacob, you are a fount of knowledge. Thank you so very much for sharing all of this and your helpful hints about how to be efficient with the computational resources and best practices that you've learned over time. Also, I'd like to thank my colleagues Ritu Tuteja and Amanda Cooksey, who worked closely with Jacob to get the apps that he demonstrated or talked about today in great shape. And again, we'll add this video recording to our playlists on genomic analyses. So head to our website or our YouTube channel to find this and many other science or technology videos online. Okay, thanks everyone for attending today. Join us in 2 weeks when we will feature the NEON Project's ecological big data resources that are available to the public for free and how to use CyVerse to exploit those for your research. Our presenters will be NEON collaborators Donald O'Leary and Bridget Haas. Until then, stay healthy and we'll see you back here in 2 weeks. Take care, everyone.
Thank you all.